All right, welcome to Roots of Reality Experiences. Today I'm joined by Dr. Russell Bicknell, who is a paleontologist and postdoctoral researcher at the American Museum of Natural History in New York City. Additionally, Dr. Bicknell's research focuses on the rise of predation and the record biological response to predators, as well as the evolution of arthropod groups and the conservation of modern forms now considered endangered. So, Dr. Bicknell, thanks for coming on today.
No, look, thank you for having me. I'm really looking forward to this.
So, just starting off, when did you first get interested in paleontology? Sorry.
So I was 5 years old once, believe it or not. And my father showed me an animal called a trilobite. It's an extinct group of animals called arthropods. And he said to me that this is an animal that's no longer alive, and the people who study these things are called paleontologists. And I was like, wow, that's cool. That's a fun thing. And then so I suppose that really was sort of what started it. And then I had a series of, I suppose, very fortunate events where I was able to pursue this pretty niche career. And yeah, that's wound me up here in New York City.
That's interesting. So, did you grow up in an area that kind of had fossils around at all, or was this like in a museum? Or
So I grew up in New Zealand, and the New Zealand fossil record is notoriously about 95% like shells, like seashells.
okay,
So this particular fossil was a gift that my father had been given, and so it was, I suppose, one of the many that he had collated. Um, so the house was actually quite full of rocks growing up. So I suppose I was surrounded by them in that way. But the New Zealand fossil record is more about sort of most recent, I suppose, geology more than anything else.
okay, interesting. Yeah, I know. Just from growing up in Kansas, my own dad used to take me around to some of the sort of limestone formations in Kansas and see all the ocean fossils and stuff there. So kind of had a similar experience there growing up, where I just constantly finding fossils around, just like off the highway and stuff and rock formations. So cool. And when did you start doing research on like horseshoe crabs?
So that was during my PhD.
Okay.
That was about 2017, 2016, 2017 is when that sort of started. And that was, so I was based in Australia at that point. So I'd moved from New Zealand to Australia. And it was a really interesting time for me because I didn't really know much about horseshoe crabs. And the focus of my PhD primarily was on the Cambrian explosion. So, like a very long period of time ago, horseshoe crabs are alive today. I was like, I don't quite see how we can reconcile that difference. But the motivation was to use horseshoe crabs as a modern comparison for what we see in the fossil record. And through that lens, I got very much into understanding the anatomy of modern day horseshoe crabs. And then the lateral extension of this, because I'm a paleontologist, was to go, well, what do we see in the fossil record? What can we understand about horseshoe crabs and their evolution over deep time? And that's really how that sort of came to be a focal point of my research.
Yeah, okay. So that's interesting. And so, how long has the horseshoe crab sort of existed? Obviously, it's certainly changed to a certain extent over time, but how similar is it to what it was, going back in the fossil record?
So I was at a conference recently, actually, and the oldest horseshoe crab has now been revealed, I suppose, and that is 480 million years old. So it's at the beginning of a time period called the Ordovician. So it's just after the Cambrian ended. The really interesting thing about horseshoe crabs is that there are periods of time in which we see animals that look very, very similar to the modern day forms. And there are also periods of time in which we see animals that we know are horseshoe crabs because they have the same structures, but they just look wildly different. And so it's this fantastic history of trying to be something that ultimately isn't a horseshoe crab and then falling back into an ecological niche or just like a place in ecology which is of a standard horseshoe crab. And that's really how they've survived over ultimately a very, very long period of time in five mass extinctions.
Wow. Okay. And so, what has, what specifically enables something to survive, like what in detail, like what is that process like? Because it seems so crazy to imagine a species that could, if you look at like the fossils of horseshoe crabs, from a person who's not trained as a paleontologist, they would say, "Wow, this is quite similar. It doesn't look like there's been much changes." So what's going on there?
So it sort of depends on when in deep time we're thinking about these animals. But if we take sort of a couple of their main evolutionary radiations, for example, to kind of delve into this, we'll do that. I preface the following in saying that I think there is a decent amount of chance in their survival. I mean, these are animals that were able to deal with, a mass extinction that killed off 95% of all marine life, that
Yeah.
outlived the dinosaurs. And so there is a degree of, I suppose, adaptability, but also just a degree of chance in the fact that they managed to survive. But focusing in on, I suppose, the big pulses of horseshoe crab evolution. So one of them was in the Carboniferous. This is a time where you have massive dragonflies, where you have massive sort of centipede-looking things, and these animals, horseshoe crabs, were mostly living in sort of marginal marine, possibly freshwater environments. So modern day horseshoe crabs don't do that. Modern day horseshoe crabs are almost exclusively marine. And what we see at this point in time is these animals just look so wildly different. They've got spines where we don't see spines today. They don't have spines at all in some cases. And we think this is to do with really filling in a whole bunch of ecological niches or just space in the ecology that was vacant at the time. But then these forms go very rapidly extinct. So by the time you get into the Permian, there's nothing left. And you go back to just a normal looking horseshoe crab. And so you're like, okay, well, they tried, they had fun, it didn't go so well. The next sort of pulse is in the Triassic. So after that huge mass extinction, almost everything dies off. We see once again horseshoe crabs living in these sort of marginal marine, possibly freshwater-like environments, and once again they just go completely nuts with their shapes. My favorite horseshoe crab of all time is called Australimulus. It's from Australia, and like all Australian animals, it just looks insane. It looks like a pickaxe, like its head's just elongated, massive spines, and everything else is kind of reduced. It's this crazy looking animal. But once again, at the end of the Triassic, these all go extinct. And so you enter into a time period once again where in the Jurassic, horseshoe crabs just look like modern day horseshoe crabs. So there is it's this really interesting story of trying to be something that they ultimately can't do, uh, experiencing extinction, but there's still populations of true looking horseshoe crabs, so horseshoe crab looking animals that were surviving, that have managed to continue on in that trajectory to leading to our modern day animals. And you're right, when you look at like Jurassic age horseshoe crabs that we know were wandering around with the dinosaurs, they look very, very similar to our modern day forms. And what we think is happening there is that there's these species, these populations are surviving within marine environments. And the only main change that we're seeing is that they are simply increasing in size. So these horseshoe crabs that look like horseshoe crabs in the fossil record are relatively small compared to our modern animals. But as you step forward towards modern day populations, we see a pretty consistent increase in the size of these animals.
Yeah. Yeah, it's interesting. I think the first time I came across a horseshoe crab was when I was in Maryland, and it was just on the beach, and I was like, "What on earth is
What is
that?"
this? Yeah.
And and, and I was like, "Okay." And then I learned a lot about 'em because I know they were in museums and people were like talking about them all the time when I was visiting the area. And truly, I mean, just some of the most interesting looking creatures I've ever seen. But obviously, a lot of people will kind of talk about them as like living fossils. What exactly is a living fossil, and if things have changed over time, does it still count as a living fossil?
So there are two perspectives on the idea of a living fossil. One of which is that it's a useful concept for sort of articulating evolutionary stasis when stuff doesn't change. The other view is sort of the view that I take in that living fossil is kind of an oxymoron because for a true living fossil to be a thing, it needs to be a species in the fossil record and also alive today. And we don't see that in horseshoe crabs. We see that sometimes in microfossils, but not
Mhm.
in these larger animals. So, but for the context of understanding evolutionary stasis of something looking very, very similar in the fossil record over deep time. It's a really useful tool. So if we think about horseshoe crabs in the context of living fossils and living fossils broadly defined, what we're talking about are species, which can be anything, like ginkgo trees are an example of this. There are an array of other arthropods like triops, that really look very similar today as animals. Lungfish, another example. These are animals that look very similar today, or plants that look very similar today, that we also see in the fossil record look very similar. And so what that's telling us in terms of sort of Darwinian evolution and ideas around evolution is that these animals have effectively not been under additional selective pressures or needs to change, such that they've maintained their shape, their form for a long period of time. And that tells us that they're probably living in pretty consistent environments. So they're not really sort of migrating into different sorts of places, and they're not really interacting with, I suppose, different predators or different predatory groups. So there aren't that many pressures, there aren't that many reasons for evolution to push them in a different direction.
Yeah. So essentially, I guess then calling something a living fossil is really just describing it as something that hasn't changed as much as other species have over time. Uh,
Exactly.
okay, that's interesting. Now, are there any species of life that exist that have barely changed at all, or is that everything changing because of the different pressures on environments and stuff?
That's a really good question. So I suppose there are things like algae that haven't really done very much, but also they don't really need to because they're not super complex,
Yeah.
and where they sit in I suppose in a food web, they're not really they're the bottom, right? Um, there is a genus of things called brachiopods. So in terms of like the order of, I suppose, how we group things, you got species, and then the level above that's genus. So there's a genus of brachiopod that they're animals that sort of look like bivalves, but they're not bivalves. But that's been around since the Cambrian. And so
Okay.
this genus has survived for, I think, 510 to 520 million years. And so there's been changes at the species level, but at the genus level, this thing is basically consistent. And so we do get those really interesting situations where these animals, they really don't change all that much. You see minute changes that be like, okay, well, that's, those two things probably couldn't reproduce together, but they're similar enough that we group them in that bigger package, I suppose.
Okay.
Yeah.
That's interesting. So, how would you get what's an example then of like a genus level change versus a species level change?
Oh gosh, that's a good question. So if we're thinking about a species level change, we I suppose if you want to make it sort of humanocentric, we can think about the fossil record of humans. And we know that that's very well filled in because we're really interested in understanding where we came from. And we know that there are other species in the genus Homo. So we are the genus Homo sapiens. And so we see that there are small changes that would differentiate these other species within the same genus from us. And that tells us that we probably would not be able to reproduce with these other species, but we're closely related to them.
Okay.
Now, if you want to think about sort of a genus level comparison, I suppose you could think, um, let's think about horseshoe crabs. And we'll take it back to horseshoe crabs. All right, so there are four species of horseshoe crab today, and there are three genera, so three different genuses. And the American horseshoe crab, the one that you saw, is very distinct from the species that we see in Asia. So it has a bunch of different characteristics on its legs, the size of it, that differentiate it from, for example, what we call the Indian horseshoe crab, the mangrove horseshoe crab. And so we know that although these are horseshoe crabs, they're very related in the grand scheme of things. They are distinct enough that we put them in those different generic groups.
Okay. That makes sense. So it just depends on how big of a difference there is between the two. Okay.
Absolutely.
Yeah. Um, and so I guess it's interesting since the species, has kind of just been around in some form or another for such a long time. What have been the predators of horseshoe crabs historically?
This is a really good question. So in the fossil record, there's very little that we can really get at in terms of understanding what has been targeting these groups, primarily because the fossils themselves are really rare. Usually we'll see one specimen. If you're lucky, you'll get a lot of them, but there aren't that many deposits where you see a lot of horseshoe crab fossils. So we're kind of limited in looking at sort of the modern systems. And in modern systems, we know that because horseshoe crabs have different life stages, so they start off as little eggs and then they sort of progress to being adults, we know that in different life stages, they have different predatory groups. So during that egg stage, they're actually quite easily eaten by migratory birds. So the red knots are a group of animals that we know at least feed on the American horseshoe crab eggs. And as horseshoe crabs grow, they go through molt stages. So they cast off their exoskeleton, they grow a bit more, and they keep going. They can be targeted by other groups. So fishes are an example. But when horseshoe crab gets to being an adult or as close to adult, when it hits that sort of really large point, there are not many animals that are willing to target that. Partly because their exoskeleton, their dorsal look helmet-like thing, is really really hard. Like that takes a lot of force to break, and partly because there's just not much to them. Like they're 90% shell, and that's just not really worth the effort of trying to digest this
Yeah.
this big spiky thing. When they're flipped over, so when their legs and their breathing apparatus, what we call the book gills, are exposed, sometimes birds can come down and peck at them and eat them that way. But for the majority of their adult life stages, they really don't have that many natural predators. And I'd say actually the majority of the damage that we see on these animals in modern systems is because of humans.
Mm-hmm.
Human interactions with these sorts of animals through fishing has resulted in probably the lion's share of the kinds of damage that we see.
Okay. So would you say then that humans are essentially their kind of biggest threat today then?
Oh yeah, I mean
Yeah.
that's probably the case for 95, if not
For almost
99% of all
everything.
animals. Yeah,
Yeah,
but yeah, absolutely.
we kind of ruined the fun for other life forms, unfortunately. Um,
Yeah.
now, what's the potential impact of things like climate change on horseshoe crab populations?
So I've actually been tackling this exact question like this week.
Oh wow, okay.
Yeah, it's at the front
Timely.
of my mind. It's kind of nuts. So we don't really know. Is kind of I mean, it's probably the case for like all animals and plants these days, right? Well, there are so many stochastic variables. There's so much chance and so many other things that feed into our understanding of how animals are responding and plants are responding to climate change that we don't necessarily know. But what we can say is as we are seeing an increase in overall temperature, we may be seeing an impact in terms of those eggs, which are really, really like important. Like those need to hatch, they need to grow to continue the species. And so there is quite the possibility that we're going to be seeing a change in egg laying and egg hatching. I think more importantly, when we're thinking about like the population level, so the bigger picture of these animals, if we see a decrease in the number or the size of females, so females are larger than males, then we are running into a really core issue of smaller females make fewer eggs. Fewer eggs mean that there's fewer offspring, and that's going to actually impact the overall dynamics longer term of these animals. And so it's important for us to think about not only how climate change can be impacting these sorts of animals, but just generally, if we're harvesting these animals for their blood and we're taking the biggest ones, then we're putting them back, and if they don't survive, then we're going to result in having smaller females, so fewer eggs, and that's going to lead to bigger problems down the road.
Yeah, so that's an interesting thing, especially when you're talking about sort of the research on blood from horseshoe crabs. Can you give a little bit of insight in terms of what's exactly going on? Because I've heard of it before, but I haven't talked about it a long time or thought about it in a while.
Yeah, so there's a sort of a historical aspect, I suppose, is important to highlight before we kind of dive into what it's really about. There was some work that was done in the 70s. It was used to determine that the blood from a horseshoe crab could be used to figure out whether or not a solution has anything bad in it, endotoxins. And this was really quite a revelation within the medical industry because previously we'd been testing like injections on bunnies. And if it's not going so well, then you'll kill off a rabbit, which is just not a fun thing to think about, really.
No.
And so there was like a new way of testing whether or not solutions that we have in injections for making sure that we don't get unwell could actually be tested. And that's quite important. So, and at the time, horseshoe crabs were kind of seen as, I suppose, being less vulnerable in the grand scheme of things. So there was a transition to harvesting horseshoe crabs for their blood and then turning effectively diluting, sorry, distilling that blood down to the chemical that they use to effectively make sure that they themselves don't get unwell, and then using that to determine whether or not solutions that are put into vaccines are pure, because we don't want to be putting nasty stuff into our bodies. It's just not a fun time. Now, the if you fast forward to maybe 5, 10 years ago, there was a push for actually making a synthetic version of this because we don't really love the idea of targeting an animal for its blood. It's just like it's just not a fun thing. And so there was a lot of research that went into developing a synthetic version of this. And the synthetic version it is pretty good, but it does have a degree of error that, of course, we don't really see in what nature produces. Because nature, if you give nature 500 million years of evolution, it's going to perfect something right down to like the most minute
Yeah.
points. But there was actually this year has been a really, really big push from an array of different, both governmental and private institutions for the adoption of this synthetic variant because the American horseshoe crab, which is what we collect for this particular chemical, is now considered endangered.
Hmm.
And so we're seeing a really important shift away from, hopefully, away from using horseshoe crabs to using this synthetic variant and refining that further such that there is a higher success rate, so and fewer false positives. And that's really, really important because we're at a stage now where if this animal is currently experiencing, was getting close to an extinction event, we want to be limiting the amount of interactions that we have with this species in that negative perspective.
Yeah. That's interesting. So like um so I guess this research then basically they were taking horseshoe crabs and the horseshoe crabs were just dying or as a result of the testing and stuff on them or
Yeah, so what the original process was, well, let's just see what is in a horseshoe crab's blood. And so a small population, I think one or two were collected, and some of the blood was removed, and I think they were put back in the wild. And then from there, after distilling it, they determined that, oh, this is actually really, really useful. And so if you fast forward to, especially during the development of the COVID-19 vaccines, a lot of horseshoe crabs were harvested, they were collected, and between a third and a half of their blood was removed. Now, if you took half of my blood out of my system, I would not have a fun time.
yeah.
I can't take like a liter being removed,
Right.
let
Yeah.
alone,
Yeah.
50% of it. And so these animals are then put back in the wild and they don't have as much of a fighting chance because their body's like, I don't have half of the stuff that I use to function. And so if you continue doing that, what happens is you have a significant die off of larger individuals. And of course, they select the pharmaceutical companies are picking up the biggest individuals, so they have the most blood. And then, but those biggest individuals are also the females. And so if you have die off of these larger females, then you have fewer eggs being produced. And that's sort of the negative flow on effect here.
Yeah. That's interesting. I'm almost a little surprised that they're even trying to release them back since they're using them so much. I'm surprised they didn't start like farming them, like, as you would with like a cow or some domesticated animal on a farm.
So, I mean, you'd think, right, that the problem is horseshoe crabs, A, don't love being in captivity, and
Yeah.
B, they take about 10 years to grow from like babies to actually
Okay.
adults. And so, the 10 years ago was the 2010s. We weren't really thinking COVID could be a thing.
Yeah.
And so, the forethought of why don't we start aquaculture in this context just simply wasn't there. And
Yeah.
also because they just don't love being in captivity in
Sure.
terms of like growing and reproducing. Yeah, it's just like the process just simply wasn't thought of. And now we're in a situation where it's like, well, it would have been nice if we had done that.
Yeah.
So now we need to focus on human developed synthetic variants.
Yeah. Yeah, definitely. I mean, because now you're at a game where, okay, since there was no preparation, now you've endangered a species,
Yeah.
and now you're trying to protect them. So, yeah, that seems like a common theme. I feel like when it comes to wildlife and uh, interactions people have with them. So you never know
Yeah.
what animal gets selected for something, and then later something happens. So,
Yeah.
unfortunately. So, what are you currently working on then beyond like horseshoe crabs and
So today, actually, I'm working on an animal called a sea scorpion. They're fossil animals. They're related to horseshoe crabs. They're fully extinct. They went extinct at that in Permian, that big extinction. And this particular specimen is from New York State.
Hmm.
And it's from a time period called Ordovician. And I'm trying to figure out what it actually is. And it's a challenge because the fossil is a little bit fragmentary. So I'm like, I think I can tell you what it's not. But so that's been my day. But other than that, I mean, my sort of my bigger picture work, I'm looking to understand more around predation broadly defined. And I do this using a tool called biomechanics. So basically, the science of understanding how things work and how things can exert force on structures. And so that's where I sort of started my horseshoe crab work, I suppose. And recently, I've transitioned to focusing on crabs because crabs are amazing. They're very, very diverse. Today they have so many different groups of crabs, and they all show this weird kind of convergence towards being like a standardized crab, for lack of a better description.
yeah.
And we have this lovely fossil record of crabs. So because crabs are crunchy, they preserve a lot better than a lot of other arthropods, and they preserve in 3D. And so we can actually take the methods that I do, these three-dimensional biomechanical tools, and we can extend them to looking at fossil crabs using our modern diversity to actually pinpoint what the fossil animals or where the fossil animals could have been in the ecosystem and what sort of animals they could have been eating as well.
So, what has surprised you most then in your research on crabs throughout
Oof.
history?
So this is really the frontier of what I'm doing. Probably the most surprising thing has actually not been like the fossil ones, but it's been the modern ones. Just like how many crazy structures these animals have. I mean, like the base body plan of a crab is not that complicated, but they have done some nuts things. Like the how they have modified their pincers to like for different like life modes and different ecologies. It's just like absolutely insane. I think some of the most insane animals I've seen, like a fiddler crab. So these are these crabs. Males have like a really, really big pincer, and then they've got like a really like tiny pincer. And so you see them a lot along the east coast of the United States. But there are a species that I'm looking at from Africa, and like its big pincer is like actually massive. Like it's almost bigger than the rest of the body of the animal, and its little pincer is like this tiny little thing. I'm like, why are you that large? What is the point in doing this? And so it's been this really like amazing period of like thinking and learning and thinking laterally about what these structures can be useful because these sorts of things they have their use in predation, but they also have other applications. And so it's been a really interesting time period for me to just go and like look at modern biology and think like why does this exist? What were the selective pressures that resulted in these sorts of structures that as someone who's spent the last I don't know decade of my life, I suppose, looking at arthropods, I am perpetually shocked at these sorts of things. And that's the frontier of science to me, and that's really exciting.
Yeah, definitely. No, I mean, Yeah, it seems like there's so much you could learn about something like that and just countless examples to study. So you should be busy for a long time.
Oh yeah, it's going to be a huge piece of work, and I'm so excited.
Yeah. I mean, there are like particular characteristics that make some crabs better than others at hunting. And
So I suppose that's more of an ecological question,
okay.
and then that kind of comes down to the sort of environment in which they're living. And for me, I'm more interested in like how effective they are at using their claws.
Okay.
And so you get these, so there's one particular group of crabs. They're called shame crabs. It's kind of sad because they have these big claws that kind of cover their face. But they've got one of their claws has like adaptations, has like modifications for actually tackling shells that have spirals, so gastropod shells, which other crabs really kind of struggle to deal with. So most other crabs, if they're kind of chowing down on a shell, they'll target a bivalve because you can just crunch it. But these animals have a means of dealing with spiral shells, which is really, really cool. And then thinking about like our fiddler crabs once again, like these are we see like smaller variants of them along close to New York City, and we see these animals that they target sort of smaller prey items, but these really large structures like these things could be tackling some pretty decent prey items, both
Wow.
soft and squishy and harder. And so it's really interesting to actually begin to question like where do these animals fit in within their ecology? Where are they in the food web? And are they, in the grand scheme of things, actually further up the totem pole than we might actually expect them to be?
Hmm. It's interesting. It's like power rankings for like a sports team, except for crabs and
Exactly.
moving on up depending on their success. So
Exactly.
cool. Well, yeah, I expect to see that'd be cool. Sometime you publish power rankings of the crabs and their
Just a
statistics.
tier list.
Yeah, that's right.
Crab tier list.
That's right. Awesome. And what's the best way for people to like keep track of your work and follow you?
So I have a Twitter account which is I think still functioning despite the current state of Twitter. But a lot of my work actually being based at the American Museum, the museum has been really supportive of putting my work out to the public. And so I'd actually say like following what the museum is putting out
Yeah.
through their various channels is probably the best way right now, actually. And because they are really supportive of researchers here in general.
Awesome. Well, thanks so much for coming on and sharing all your knowledge. And yeah, definitely a cool subject that I think people may have heard something about, but don't know a lot about it. So really cool to get you on and talk about it. So
And thank you very much for making the time. I mean, I love talking about this stuff. It's my career, it's my life. And if I can have introduced a new idea to someone today, then I'm more than happy to have done that.
yeah, absolutely.